Hello, my name is Scott McElveen, and this is your hyper-efficient BeCrop Farm demo. So as you know, soil has three dimensions to it: the biological, the physical, the chemical. We've had great soil tests for decades for the physical and the chemical aspects of soil performance, soil behavior, soil health. The biological has been neglected in our farming. For the most part, for the very non-nefarious reason that there were not good tests for it, not high resolution, agronomically relevant, affordable, and actionable tests. So this is where BeCrop comes in, to that component, filling in that blind spot so that we can have 360 holistic soil management. What do? We extract DNA from soil. We snip out just the marker gene, what you could think of as a fingerprint to just identify the microbe. The cost of soil DNA testing has come down massively in the last few years. Using this fingerprinting approach brings down the cost even more, and it's made possible by the fact that we've built the world's largest database of that fingerprint data, those taxonomic references for specifically agricultural microbes. That way we identify who's present, and then thanks to a very large reference database of the full genetic potential and abilities of a microbe, we're then able to say, okay, does it promote plant growth by producing plant growth hormones? Does it liberate crop available nutrients? Does it immobilize, lock up crop available nutrients? Does it cause disease, prevent disease, so on and so forth? Hey, quick timeout from future Scott just to say, please don't be misled by my DIY screen cap approach to this demo. Biomakers is a brilliant, multidisciplinary international team. We've been in operation for 10 years. We've worked with everyone from industry giants like Bayer, Corteva, Syngenta, Nutrien, to smaller companies like TMAC Agro, InSoil Algae, Huma, to individual growers and independent crop consultants. So I did this little demo from my desktop so that I could focus on the details that I think are most important to smush into a compressed 10-minute demo. But don't let that fool you. We have a very strong company behind the technology I'm demoing here. Any BeCrop test, any BeCrop technology is going to offer you information in these categories: your NPK and micronutrient cyclers, your carbon cyclers, disease-causing organisms, organisms that suppress fungal and bacterial disease, as well as root-feeding insects and nematodes, plant growth promoters, stress-mitigating microbes, overall biodiversity, functionality, resilience, connectivity of your living soil community. Excellent information that's been delivered for the last 10 years as a PDF report with all kinds of data about agronomically meaningful and influential biological functions. We've also produced wonderful tools that I use every day in our portal for comparing samples or groups of samples to figure out biological drivers of crop performance issues, ways to line up samples and look for patterns, commonalities, and disparities between samples and sample groups, looking at the specific microbes within a sample. Now, this is, to me, as someone who's trained in microbiology, this is absolutely revolutionary in terms of characterizing the living community and soil. Nevertheless, this is an unwieldy test for many growers and agronomists because it wasn't clear how to what was the spatial variability across a landscape? And there's no recommendations to this test. Lots of good information, but what do I do with it? What's the actionability? How do I influence this without, it asked a lot of people to become educated and do experimenting to understand how they can influence these metrics. And that called into question or, made less valuable, less clear the value of the testing. And so let's tackle that first question because it enabled us to knock out the other two. How do we characterize a landscape? Well, what we're able to do is we can take in shapefiles for a field, and we analyze that landscape on the basis of spectral data, that is satellite imagery. What is the crop telling us? What is the crop telling us about its nutrient uptake, about its chlorophyll content? We're looking at NDVI, leaf area index. Spectral data can also tell us about soil moisture. It can tell us something about the soil itself. Other environmental data, we can learn about soil type and slope and aspect, precipitation. Over 1,000 parameters go into this analysis by which we're able to cluster the soils, basically create biological zones for the farm. And on that basis, we can strategically assign where are we going to sample. And we've found that doing this, we can decrease the necessary sample load to get adequate precision, accuracy, and resolution for making meaningful, informed management decisions. And what that looks like is, let's hop into an example, looking at nitrogen, for instance, and I'm going to keep this brief, high level. If you want to learn more, please get in touch. We can do a more in-depth demo. But let's just look at our organisms that can release crop-available nitrogen, that can mineralize nitrogen. This field, low capacity to do so. This bright yellow is in the bottom bracket, very low. We've got a 15 here. That means on the scale of what has been observed by our test in the last decade of testing, which has involved a lot of corn, this is on the 15 out of the 0 to 100 scale of what is possible, what is conceivable, what is observable in corn. These fields are low, but substantially better off than the 15. Then if we look at our soil's tendency to lock up available nutrients, that same field that had the lowest capacity to release crop available nitrogen also has the highest tendency to lock it back up again, to immobilize it. And so we have a soil here that is not working for our crop, but rather against our crop. And so therefore, this is also the field that will be most responsive to interventions to improve nitrogen availability and uptake efficiency. And we can talk about, of course, if you get in touch, what those strategies are, because they range from the cultural practices, placement and rate and source to more explicitly biological solutions. Similarly, if we pop over here and look at our phosphorus solubilizing microbes, all right, we know that fertilizer use efficiency for phosphorus is abysmal in general. Well, this field, it already has a relatively strong capacity to biologically solubilize locked up phosphorus and is therefore less likely to respond to a biological tool in order to enhance phosphorus solubilization, at least a living one, whereas a phosphorus solubilizer more likely to show response on this field where that capacity natively is lacking, is inadequate. There's nutrient cyclers, N, P, K, 8 micronutrients. Here you have a good many of them lined up. We have our carbon pathways. These can help inform us about our capacity to break down crop residue, to mineralize nutrients. They can tell us about the oxygenation in our soil. Our disease metrics, let's take a look at red root rot. We can see where the hotspots are, where the populations of pathogens are most abundant, where we need to focus our management, where we can scout most effectively. Go to the areas that are most likely to show symptoms, most likely to be experiencing yield loss, lost yield potential due to a pathogen. This also enables us to more proactively, more preventatively manage disease instead of being reactive and more targetedly. Crop protection can be costly. This can enable us to target and test alternative solutions, such as biocontrols. We can also see our native capacity to control diseases. Fungal, relatively strong capacity across most of the field. Capacity to manage bacterial diseases like Gauss's wilt here, low, among the lowest we've ever observed in corn. We have our plant growth hormone producers, our stress mitigators, overall indicators of the capacity to build carbon or liberate nitrogen, phosphorus, potassium, overall capacity to control disease, adapt to stress. We can look at all those categories I showed you before. They're here and mapped in order to be efficient with time. I'll show you there are field level reports where you can look at each field, the variability, where's my optimal zone. We also have a farm level report, which will show you here are the interventions based on our trials database, world's largest database of biological product efficacy. I don't know why the report is not loading in right now here. In case this happened, I've got it screenshotted onto a slide over here. The first page of it's got several pages, including a summary of the disease risks, but here it'll say, based on our trials results, matching a trials location to the field conditions on this field, to the biological risks and opportunities on this field, what are some broad third-party trials-backed, brand-agnostic active ingredients that would be effective, most likely to be effective based on our trials at improving nutrient cycling bottlenecks or overall soil collaborativity, cohesion, ability to build soil structure, mitigate stress, promote growth. And so without going into any further detail, I'm close to the 10-minute mark that I'm aiming at. Here's a quick look at, nitrogen cycle health in April can be predictive. This biological component can be predictive of overall biomass accumulation several months later into the season. So please feel free to reach out. There's a whole lot more to say, a lot more to show. We're working on integration with precision ag platforms so you can have a prescriptive map that takes into account not just your nutrients, but your nutrient cyclers. So don't hesitate to get in touch. We'd be more than happy to work with you, with your growers, and enable you to advise well on full 360 soil health, soil performance. Thank you very much.